Hi, I'm Eric Haura. I'm the director of the Lung Cancer Center of Excellence here at the H. Lee Moffitt Cancer Center. And we're here today for our 5th Behind the Science series. And my guest today is Dr. Alvaro Monteiro, who is a scientist in our center. And today we're going to learn about the mechanisms of how chemotherapy works, how chemotherapy damages DNA, and why this is important for producing cures in patients with lung cancer, and really more importantly, how the science here can make chemotherapy work better. So Alvaro, thanks for joining us today.
Thank you.
Why don't you tell us a little bit about your background?
Yeah, my background is in genetics primarily, and then I did a postdoctoral training in molecular biology of oncogenes, genes that were involved in cancer, and my career has been mostly focused on genetic factors that are important for cancer, cancer predisposition, cancer etiology and treatment.
I see. So for many years we've used chemotherapy to treat many different types of cancer, including lung cancer. You know, in general, some people can do well with chemotherapy and some people can do poorly with chemotherapy, both in terms of the outcome, meaning does the tumor shrink, and what their side effects are. So explain to us how chemotherapy is thought to work, and what your general thoughts are on how we can actually make it better, either make the tumors disappear more or alleviate some of the side effects.
Okay. You know, a large class of chemotherapy drugs are drugs that are primarily geared to damaging the DNA. And the idea here is that if you damage the DNA of the cancer cell hard enough, then the cell will eventually die. The problem is that whenever you're doing something to the cancer cell, the normal cells that are all around your body are also going to feel the pain. So the critical thing is to find that right amount and that right class of chemotherapy that can kill the cancer cell without killing the normal cells. And in a way, that's why we study what we call the DNA damage response network. So if you will, you can think of a cell as a city in which the DNA damage response network is everything in the cell that is what we can think of our first responders. The fire department, the paramedics, that every time there is a problem in the cell, these proteins will come in and try to fix it. When this communication between these first responders is defective, then that leads to cancer. We know this because we know that in many people that inherit defects in the DNA damage response network, they actually have familial cancer syndromes that are very important. But this is one part of the story, the part of the story that actually defects in this system leads to cancer. But we can also use these defects, if we know exactly what they are, to exploit treatment. So basically the idea is that these cells, the cancer cells that had a different rewiring of how these first responders communicate, we can actually now find ways in which these cells are now more susceptible to chemotherapy than the normal cells that are all around them.
So you mentioned a word called networks. So, you know, probably most people are familiar with, you know, social networks or, you know, Facebook networks. But in the last couple of years, there's been this push for network biology. So why don't you tell us a little bit about, you know, what is this general approach of network biology? What does it mean? What is the power in terms of being able to help with patients ultimately?
Okay, so you know, you made the connection with the social network, and it's very similar to a social network in which each protein in the cell will talk and interact to other proteins in the cell. And some of these proteins are very popular and they talk to many people. Some of these proteins are not that popular and won't talk to many people. And the key is really to find out who talks with whom at what time.
I see.
So this is very important for the network. And it's very similar to what we have for social networks.
So with this analogy of like social networks, I imagine the protein who's equivalent to a very popular person or a big talker, someone who's doing all the communication, would be maybe a good protein to go after?
Yes, but not without its problems, right? So if you continue the analogy with the social network, you can look for people that have many interactions and say that person is very important. But we have what we call the pizza delivery guy problem, in which if you are trying to map a person that has contact with many people in one neighborhood, that's the pizza delivery guy.
Yeah.
But you can argue that if you take the pizza delivery guy out of this community, really people can get by without pizza without any problem. So it's a little bit difficult to find out exactly which one we're going after, but we try to do this in a systematic way.
So you
Responders look
essentially made a map of the social network of all proteins, or a group of proteins that were the first responders of DNA damage.
Exactly. So if you want to take the analogy a little further, imagine that you have a switchboard for all the 911 calls, and some goes to the fire department, some go to the water management, some goes to the paramedics. So what we try to do is, in the cancer cell, we find out exactly where this communication goes.
So what's your ultimate dream of how this research could ultimately benefit lung cancer patients? If things went according to plan and perfectly, but what's your vision of what this would bring?
So the vision here is basically to have a very detailed map of how these things can interact. And once a patient comes in the clinic, the tumor and the patient would both be examined for their genetic profile. That genetic profile will now be overlaid into this map of interaction. So now we create a personal network.
I see.
With the personal network that basically you can take to any of the clinicians that are treating you, you can now predict much better what will be your response to different drugs and what perhaps will be your outcome. So the idea would be to have a dashboard that can be used to look at the status of your disease and how we can basically use these instruments and this information to give you the best treatment at the best time.
Yeah. You said one key thing, which is that you'd be essentially making these dashboards for, like, normal cells based on who you are as a person. So I mean, a lot of people have concerns about the side effects of chemotherapy. It seems like the promise of this approach too would be to inform the clinician and the patient about the side effects. Alvaro, thanks for joining us today. I know I learned a lot. I think the major things that I found in the discussion today is chemotherapy is still a very important tool for treating lung cancer, both in people who have very advanced lung cancer or even in people who had surgery and we're trying to prevent the recurrence of lung cancer. And so it still has an important role, but it needs much more improvement, both in terms of curing more people with lung cancer as well as finding better ways to deal with the side effects. And I'm very encouraged by this research as being able to point us in the right direction and ultimately lead to better outcomes. So I thank you for joining us, and we'll see you next time.